This is Fred, and today Fred has just been diagnosed with HIV. So for that reason, Fred is really sad. Fred is one of 37 billion people in the entire world to have this unique virus. In the 1980s, HIV really rose to prominence, and this was the first time that humans actually found a virus that we couldn't really cure. It didn't affect one country or one race, but it affected the entire world. So why is HIV such a serious illness today, and what makes it different from any other illness? HIV stands for the Human Immunodeficiency Virus. What makes this different from any other virus is that it affects the human's immune system. The body's defenses that are used to eradicate diseases in our body is targeted, and therefore the immune system can't even target the HIV virus infection itself. The virus infects the blood and the immune system until a majority of the blood is just the virus. HIV targets white blood cells until the entire white blood cell count is depleted. Like most viruses seen today, HIV is made up of proteins and a nucleic acid, which is specifically a single-stranded RNA strand. HIV differs because it has a specific enzyme known as reverse transcriptase, which can reverse transcribe its RNA into DNA and later integrate that DNA into the cell's DNA in order to make more copies of itself. The proteins that make up the HIV virus have a similar structure to any protein, with a carboxyl group, and an amine group, and an R group. Within that R group, the R group can be either polar or nonpolar according to its molecular properties. Just like we learned in class, the intermolecular forces within these proteins dictate its structure and therefore its function. HIV has a specific protein known as GP120. GP120 is a glycoprotein. This protein is located on the outside of the HIV virus and coats the nuclear envelope. A glycoprotein means that it's made of both carbs and proteins, and it is used for cell recognition and identification for the virus. HIV affects the white blood cells in our bodies, but most specifically, CD4 plus cells. These are white blood cells that have CD4 receptors on its surface. Because HIV affects these cells, the CD4 cells are not able to do their job and defend the entire body and the immune system, so therefore the immune system becomes deficient. The HIV virus is able to get into the cell when the GP120 protein attaches to the CD4 receptor on the outside surface and the co-receptor inside the cell. These co-receptors vary based on the type of CD4 plus cell. In order for the virus to get into the cell, the GP120 protein must interact with both the CD4 receptor and the co-receptor. These proteins interact based on their molecular makeup in the R groups. These R groups can be either polar or nonpolar like discussed before, and this enables the virus and the cell membrane to change shape, and therefore the virus fuses with the membrane and the nuclear material goes in. Once the virus is inside the cell, the single-stranded RNA retrovirus is able to use reverse transcriptase to change the RNA into DNA. This DNA is integrated with the cell's DNA, and when proteins are transcribed, immune response proteins are made, as well as the virus, which is able to infect the rest of the body. The progress of disease progression in an individual can be studied on this graph. The blue signifies levels of the CD4 white blood cells in the blood, and the red signifies the concentration of HIV virus in the blood. This spike in HIV concentration signifies the acute stage in HIV disease progression. This means that the virus is cranking out versions of itself very quickly, and the body responds with flu-like symptoms. The next stage is known as the asymptomatic stage, in which the virus slows in production but exists. This stage can last up to 1 to 9 years, and slowly but surely, the HIV virus depletes the immune system. The last stage on the curve occurs when the HIV virus completely overruns the body's immune system. At this point, most of the body's white blood cells are killed off, and the body is no longer able to fight off simple infections that would otherwise been fought off if the body was not affected by HIV. At this point, the individual is considered to have AIDS, or the acquired immunodeficiency syndrome, and is likely to die within a few weeks.
In order to understand the way in which the body can be infected with HIV, it is important to understand the way in which the immune system works. The immune system is the body's defense against infection, and it is comprised of the innate immune system and the adaptive immune system. The innate immune system is a set of defense mechanisms that we have from birth. These include physical barriers, such as hair, skin, and nails, and other mechanisms such as bodily secretions with lysozymes to expel intruders, and inflammatory response, which is when blood rushes to the site of infection to bring immune cells to the infection site. The immune cells that make up the innate immune system are phagocytes, macrophages, mast cells, neutrophils, eosinophils, basophils, natural killer cells, and dendritic cells. These cells clear the body of infection by eating or engulfing the invader. The other part of the immune system is the adaptive immune system. It is activated by exposure to different pathogens and uses immunological memory to precisely enhance and attack specific intruders. It uses B cells and T cells, which are lymphocytes, to perform these attacks. T cells are responsible for destroying invaders. There are different kinds of T cells, such as progenitor T cells, helper T cells, cytotoxic T cells, and regulatory T cells. T cells attack pathogens with surface receptors that allow it to bind to specific antigens. They are comprised of alpha and beta peptide chains held together by a disulfide bond. It binds to these antigens via hydrogen bonding, electrostatic interactions, and hydrophobic and van der Waals forces between the receptors and the antigens. Once it binds to an infected cell, it can release cytotoxins to kill it. B cells are responsible for sweeping up the parts of the pathogens that are left over after the T cells attack. T helper cells activate B cells, which can become plasma cells, which produce immunoglobulins, a type of antibody, which are large proteins that bind to the surface of foreign bodies, to come to the region and attack it. B cells can also develop into memory B cells that can then remember how to attack the same thing if it invades the cell again.
Because there is no certain cure for HIV, scientists have developed a class of drugs known as antiretrovirals, which inhibit specific enzymes like protease and integrase that play an important role in HIV infection in the cell and propagation throughout the body.
Protease inhibition prevents the release of individual core proteins and the overall maturation of the virus. The binding of the inhibitor is reversible, but blocks the polyprotein from entering the active site to be cleaved. The inhibitor mimics the substrate of protease, but where there would normally be a peptide bond, instead there's a hydroxyethylene group. Protease has difficulty cleaving the hydroxyethylene due to its hydroxyl group. Normally, the carboxylate groups of the enzymes aspartic acids would cleave the substrate. However, these hydroxyl groups form hydrogen bonds with the carboxylate groups instead, inhibiting their function. There are multiple ways to inhibit reverse transcriptase. One way is through the use of nucleoside reverse transcriptase inhibitors, or NRTIs. They target a specific binding site in the enzyme for competitive inhibition with a normal substrate. Most inhibitors are comparable to deoxynucleosides that are normally used to form DNA but may lack the hydroxyl group found on the third carbon of the deoxyribose, and instead have some other molecule. The chain is terminated because a condensation reaction cannot occur to properly form the backbone of the DNA. An example of an NRTI would be zidovudine, or AZT. It is the same shape as a thymidine molecule and is incorporated in the forming DNA strand in its place. Unlike thymidine, AZT has an N3 molecule where a hydroxyl group would be, stopping the chain since the hydroxyl group is necessary for the dehydration synthesis of DNA.
So, Fred, you can finally wipe those tears off now that antiretrovirals exist. Antiretrovirals work, and so that the infection is slowed within an individual, and individuals like Fred are able to live happier and longer lives. So, Fred, because of drugs like zidovudine, you can live a happy and healthy life with HIV.